Proteins and enzymes, the catalysts of life processes. Proteins and enzymes are essential components of living organisms that play a significant role in life processes. Proteins are complex molecules consisting of amino acids, while enzymes are specialized proteins that catalyze chemical reactions in living organisms. This article will explore the structure and function of both proteins and enzymes, their classification, and their role in the various life processes. Structure of proteins. Proteins are made up of amino acids, which are organic molecules containing an amino group, NH2, and a carboxyl group, CoO. There are 20 different amino acids that make up proteins, and they are joined together in a specific sequence to form a polypeptide chain. The sequence of amino acids in a protein defines its three-dimensional structure, which determines its function. Proteins exist in 4 different levels of structure: primary, secondary, tertiary, and quaternary. The primary structure of a protein is its linear sequence of amino acids. The secondary structure refers to the folding of the amino acid chain into alpha helices and beta sheets held together by hydrogen bonds. The tertiary structure results from the folding of the secondary structure due to interactions between amino acid side chains or groups. The quaternary structure is the final structure resulting from the association of 2 or more tertiary structures. Function of proteins. Proteins have a wide range of functions in living organisms. They act as enzymes, hormones, transporters, receptors, structural components, and antibodies. Enzymes are proteins that catalyze chemical reactions in living organisms by lowering the activation energy required for the reaction to occur. Hormones are signaling molecules that regulate various physiological processes in the body. Transporters are proteins that facilitate the movement of molecules across membranes. Receptors are proteins that recognize and bind to specific molecules, transmitting signals to initiate various cellular responses. Structural proteins provide support and shape to cells and tissues, while antibodies are proteins that help the body to defend against infections by recognizing and neutralizing foreign molecules. Classification of enzymes. Enzymes are a special class of proteins that catalyze chemical reactions in living organisms. They are classified based on their function, which is determined by their structure and the type of reaction they catalyze. Enzymes can be divided into 6 main categories based on their mode of action: hydrolyses, isomerases, ligases, lyases, oxidoreductases, and transferases. Hydrolyses are enzymes that catalyze the hydrolysis of molecules by breaking a bond using water. Examples of hydrolyses include lipases, proteases, and nucleases. Isomerases are enzymes that catalyze the conversion of molecules from one isomer to another. Ligases, also known as synthetases, are enzymes that catalyze the joining of 2 molecules together using energy from ATP. Lyases are enzymes that catalyze the breaking of a bond in a molecule without requiring water. Examples of lyases include decarboxylases and dehydratases. Oxidoreductases are enzymes that catalyze the transfer of electrons from one molecule to another. Transferases are enzymes that catalyze the transfer of a functional group from one molecule to another. Catalysis of enzymes. Enzymes catalyze chemical reactions in living organisms by lowering the activation energy required for the reaction to occur. This reduces the amount of energy required to initiate a reaction and speeds up the rate of the reaction. The active site of an enzyme is the part of the protein that interacts with the substrate and facilitates the catalytic reaction. The active site is specific to the substrate and facilitates an induced fit, where the enzyme changes its conformation to accommodate the substrate. Enzymes can also be affected by various factors that affect their catalytic activity. The pH of the environment, temperature, and concentration of substrates and enzymes are crucial factors that affect enzymatic activity. Enzymes have an optimal pH and temperature range, where they exhibit their maximum activity. A change in the pH or temperature outside this range can denature the enzyme, rendering it inactive. Role of proteins and enzymes in life processes. Proteins and enzymes are involved in various life processes such as metabolism, DNA replication, protein synthesis, and signaling pathways. Metabolism refers to the process by which cells break down or build molecules to generate energy or create new molecules. Enzymes are essential in catalyzing the various metabolic reactions occurring in living organisms. DNA replication is the process by which cells copy their genetic material to produce new cells during cell division. Enzymes such as DNA polymerase catalyze the addition of nucleotides to the growing DNA strand, while DNA ligase joins the fragments of the DNA strand together. Protein synthesis is the process by which cells create new proteins using the genetic information stored in DNA. Enzymes such as RNA polymerase catalyze the transcription of DNA into messenger RNA, mRNA, while ribosomes catalyze the translation of mRNA into proteins. Signaling pathways refer to the complex network of molecular interactions that allow cells to communicate and respond to external stimuli. Proteins such as receptors and signaling molecules play crucial roles in these pathways, facilitating the transmission of signals between cells. In addition to their essential roles in life processes, proteins and enzymes have important applications in various fields such as medicine, food science, and biotechnology. In medicine, enzymes are widely used as therapeutic agents for the treatment of various diseases such as cancer, HIV, and cardiovascular diseases. In food science, enzymes are used in food processing to improve the texture, flavor, and nutritional value of food products. Enzymes such as amylases and maltases are used in the production of bread, while proteases are used in the production of cheese and beer. Enzymes are also used in the production of sweeteners such as high fructose corn syrup, and in the clarification of wine and fruit juices. In biotechnology, enzymes are used as tools for DNA manipulation and genetic engineering. Enzymes such as restriction enzymes and ligases are used in recombinant DNA technology to cut, paste, and join DNA fragments to create novel genetic constructs. Enzymes are also used in the production of biofuels and bioplastics, where they convert biomass into useful products. Moreover, the study of proteins and enzymes has significant implications in various fields of research, including biochemistry, molecular biology, and genetics. Scientists have been conducting research on the function and structure of proteins and enzymes for several decades, and new discoveries are being made regularly. In biochemistry, proteins and enzymes play a critical role in understanding the chemical reactions that occur in living organisms. Scientists use biochemical methods to study the interactions between enzymes and their substrates at a molecular level, providing insights into their catalytic mechanisms and regulation. In molecular biology, the study of proteins and enzymes is essential in understanding the genetic code and how it is expressed. In genetics, proteins and enzymes have been extensively studied for their role in genetic diseases. Genetic mutations in enzymes can lead to metabolic disorders such as phenylketonuria and Tay-Sachs disease. Similarly, DNA mutations in some enzymes that regulate DNA repair can lead to cancer. Scientists continue to explore and discover new proteins and enzymes, uncovering their functions and potential applications. For example, researchers are studying enzymes that can break down plastics, hoping to develop new solutions for the problem of plastic waste pollution. In addition to their roles in biological processes and various fields of research, proteins and enzymes also have cultural and historical significance. For centuries, humans have used enzymes in food production, such as the fermentation of wine, beer, and yogurt.